Okay, so what we are going to learn today is that we start our lives from single cell after the fertilization. So sperm cell fertilizes the oocyte, and that's a single cell which becomes diploid. Okay, now after fertilization, this cell now undergoes mitosis. One cells go to two. Each of this cell is further undergoing mitosis. So this is how we grow. An important point or a question to understand is that each cell, and we can, this keeps going like this, mitosis, successive mitosis. And then eventually we have different kinds of cell types. For example, we have skin cells, we have muscle cells, we have neurons, we have blood cells. Within blood, there are so many different cell types within blood. You have red blood cells, you have white blood cells, you have platelets. So we humans have more than 200 different cell types. More than 200 different cell types. Now the question is, each cell, all of them are progenitors of this cell, which means each cell contains same 46 chromosomes, except cells which undergo meiosis. And when they differentiate, these cells which make up, skin tissue or muscles, etc., they all contain 46 chromosomes, which means all the genes which are present on these 46 chromosomes, they are present in all the future progenitors of this particular cell. Then, how cells become different? If same set of genes are there, same chromosomes are there. I mean, if same chromosomes are there, obviously then it means same genes are there. How cells, they become different from each other. And a phenomenon which we call differential gene expression, differential gene expression, that explains how they become different, which means differential gene expression means if I say A to Z is the total collection of genes on these 46 chromosomes. Now, if we say on this side we write different cell types, out of these 20,000 genes which are present in a single cell, sprinkled over these 46 chromosomes, not each cell is expressed in, not each gene is expressed in every cell type. So for example, A may be on here, off there, on here. Then B may be on here, off, on. C may be off, on. So in 21st century, what we are trying to see, what is the cell type specific gene expression program, cell type specific gene expression in different cell types. For example, in skin, epithelial cells, skin mesenchymal cells, uh, platelet cells in blood, or neurons. Then there are different kinds of neurons. So I oversimplify this, and I mean, the idea is we have a code like 1010, like a binary code. But biology does not really work like this in such binary code, but that's what our dream is. Okay. So how they become different? Not all 20,000 genes are regulated like this. You should know we have genes which are called housekeeping genes, and 70% of our genes are like housekeeping genes. These are the genes which are constitutively active in all different cell types. And then there are tissue specific or developmental genes. In fruit flies, we say there are nearly 200 developmental genes. They are so crucial. If one of the 200 genes get mutated, fly dies. It's that important. Now, we say if all the somatic cells, let's say this is skin cell, let's say this is a blood cell, and then this is a neuronal cell, which takes away, messages from brain to muscles, etc. If all of them contain 46 chromosomes, starting from the fertilized egg, how? So one thing we said, through differential gene expression. What did we say? Through differential gene expression, cell type specific gene expression patterns are established. Early on, cells which will be committing to eyes, they will start expressing eye specific genes and switch off all the non-essential, which are not going to contribute to eye. But the question is how all this is achieved. And in 21st century, we say epigenetics is involved. The DNA sequence of all the genes on 46 chromosomes, it's not at all altered. It's not perturbed at all. It remains same. But a level above genetics, which is what is the basic genetics? DNA sequence in the chromosomes. So there's no change at all in the DNA sequence, but a level above, which can be the nitrogenous bases or histone proteins. These two, they get modified. They get chemical modifications, which are covalent in nature, chemical modifications. What's wrong? Huh? Why these slides are not showing anything?
So Control Z करके try करो.
Edit no. Control Z is also not working. What went wrong? Huh?
सर आपने कैंसिल तो नहीं किया था when it asked you if you want to keep the note annotations?
No, I just clicked so that I go to the next slide. Anyway, that's not important. Why I cannot change the slides? Okay, we can start again, all over again. Hmm. It was such a good lecture. It was going, and all of a sudden. Okay. Apparently, I have lost my whatever was going on. Anyway, let's move on. So what we were saying that it's the epigenetics. And this epigenetics is modifications, chemical modifications of, for example, one of the most common modification in eukaryotes is cytosine modification, cytosine methylation. The cytosine, it gets methylated, methyl group at 5th position. And if cytosine exists in such a sequence context, and we call this symmetrical sequence because it's a palindrome to go 5 to 3, and on the opposite strand also, so it's a palindrome. So this cytosine, this lollipop represents methylation. If it is in the promoter region, gene gets silent. Silent means it's turned off. No more transcription is taking place. Opposite of silencing is activation, gene activation. And what they discovered that there are specialized proteins which are called DNA methyl transferases, which catalyze this modification and cause silencing. Now, what is special about methylation that when you will have, I'm not writing cytosines now, when you will have symmetrical methylation, when DNA gets replicated to the daughter cells, it's inherited. So following the semi-conservative DNA replication, so this mark, it persists through DNA replication and mitosis. At the DNA replication fork, right at the fork of DNA replication, an enzyme called DNA maintenance methyl transferase, MTS, methyl transferase. What it does, it recognizes hemimethylated strand and methylate the opposite strand where symmetrical cytosine is there. You can imagine now this cell will replicate, and again, these methyl patterns will be inherited. So it means it's not only the basic genetic code which is being inherited, but the epigenetic modification. We call this epigenetic modification. Why epi? Epi means above. Above the genetic code, this modification behaves as a genetic entity. It gets inherited just like DNA is being inherited. So this we call epigenetic inheritance. Now, in addition to DNA methylation, so this was what? DNA methylation in eukaryotes. It persists in plants, it's present in mammals, but very little to rudimentary in flies and yeast, nothing, okay? However, if we said in the beginning, not only DNA, but other proteins which are packaging the DNA, the histones, they also get chemically modified. H3, H4, H2A, H2B, and then we learned that there are 2 rounds of DNA 146 base pair. If I just try to draw the histone H3. Histone H3, so these are the N-terminal tail. All the histones have unstructured amino terminal tail. So amino acids one, the amino acid 1 of histone H3, and then so let's say here's an alanine, then arginine. I'm just randomly writing arginine, then lysine. So you can keep going like this, different amino acids. These proteins are histones, they are highly basic in nature. Histones are highly basic in nature. They contain basic amino acids. These amino acids, for example, lysine at position 4, lysine at position 9, lysine at position 27, lysine at position 14 in histone H3. Okay, this is specifically histone H3 we are talking. So lysine 4 can be methylated. Lysine 9 can also be methylated. 27 can also be methylated. Lysine 14 can be acetylated. Methylation can be mono or di or tri because of the, chemistry of the lysine side chain, but there can only be a single acetylation. Different modifications are there. For example, if histone acetylation is there, genes, it correlates with active genes. Genes are being transcribed. But if we have lysine, K is lysine methylation, its sequence or its context dependent. K4 trimethylation correlates with active genes. Genes are being transcribed. K9 trimethylation correlates with constitutive heterochromatin, which never gets transcribed. Constitutive means, it's a gene poor region. Very few genes are there, but if genes are there, they are permanently switched off. Lysine 27 trimethylation is also it correlates with silent genes, but this is not constitutive. This is developmental gene expression. And 2 proteins, polycomb group and trithorax group. Polycomb group proteins are silencers. They recognize trimethylated lysine and block transcription, helps in switching off genes. Trithorax, on the other hand, it counteracts the polycomb silencing. It catalyzes and recognizes lysine 4 trimethylation and switches on genes. So this is just basic intro about how genes get differentially regulated. Let's come back to our early developmental stage. We have learned, some of the mechanisms, let's say polycomb silencers, trithorax activators, these different histone modifications or DNA methylation we talked about. All these are different mechanisms or tools in the arsenal of a cell to differentially regulate genes. Now, if we were fertilized and we go to single cell and the mitotic phase, we go to 2 cells. Tell me how many of you have seen the video I uploaded as well. Did you see the video on LMS?
Yes, sir.
Good. Only 2 people or more. So watch that video so that you understand the significance of the differential gene expression, because we start our life from single cell and we have to generate more than 200 different cell types. So after cell division in mammals, invertebrates, there comes a stage, a developmental stage, which is called blastocyst stage. Embryo, at this time, it gets clearly separated into 2 different kinds of cell types. One is called inner cell mass, the other are called trophoblast, or trophectoderm, cells. The trophoblast or trophectoderm cells, they are the placental cells. They are the ones which get us connection with our mothers in the uterus. And the inner cell mass are all the embryonic cells. All the cells which are present today in our body, they are actually driven from ICM. And in that video, which you must have noticed that, cells, they start moving. The embryo undergoes different changes. So this stage is called blastula or blastocyst stage. And after blastocyst stage, what happens? Cells, they start moving, and there are 3 different layers of cells which are called germ layers. They are different than the germ cells. Don't confuse them with the germ cells. Germ layers, so cell after blastocyst, when they start moving, large scale movement of the cell start, that stage is called gastrula or gastrulation. And 3 germ layers which are produced as a result of gastrulation movements are called ectoderm, mesoderm, and endoderm. This one makes your skin, endoderm makes your gut, your inner organs, etc. So already at this stage, which is hardly few days after fertilization, we have 3 different cell types within the same. We cannot distinguish them. We cannot see them at all. Morphologically, we cannot distinguish them. But these 3 cell types, if we take out their total RNA, all the genes which are being transcribed, and we try to analyze, we will find already 1010 code, which means differential code for let's say ectoderm, meso, and endoderm. Clear? The epigenetic phenomenon, actually, this is where the epigenetic phenomenon kicks off already. Genes which are silent, which are not essential for ectodermal cells, they will be switched on. DNA methylation, talking with cell signaling, and histone modifications, they start playing a role. And it goes for all 3 different cell layers. Is it clear so far?
Yes, sir.
No question so far. Why?
Sir?
Yeah.
Sir, can you explain the blastocyst stage Walla cycle, the one that you assumed in the video? Okay, we already know that cycle.
No, this is just cell division. So cells keep dividing. So you have a single cell. After going through many successive quick mitosis, And these early divisions are called cleavage divisions. Cleavage divisions. Early mitosis. They don't involve any growth of cells. Cell just DNA replication division, DNA replication division. The overall size of the embryo does not increase. And then there comes a stage where, here cells first, the identifiable stages are, the 8-cell stage, 8-cell stage, then comes 16-cell stage, and then 32, and eventually we see a big cavity. So this big cavity is in center, and there's fluid. This is a fluid-filled cavity, which is called blastocele, C-O-E-L, blastocele. And all the cells are organized here, and this cell, all the cells in embryo are divided into 2 clusters. One are the trophoblast cells, which are going to get us connected with our mother through this cord, the placenta. And the other cells, the inner cell mass, are the cells which are going to make all of us, the proper organism, the human baby or any other mammal. Clear?
Yes, sir. Thank you.
Thank you. So now what we have learned over last 30 years in particular, that epigenetics, that brings us the answer how genes are differentially switched on and switched off so that cell fate determination is possible. The word is beautiful. Cell fate determination. Fate of every individual cell is being determined at this particular stage by differentially regulating gene expression, by differentially switching on, switching off those key developmental genes, 200 or so, which causes cells to commit, and the word is commitments, commit to different fates. Once cell fate determination takes place, cell fate determination takes place, each cell, it keeps dividing mitotically. These arrows indicate successive mitosis. And what we see eventually, the end result is we see, morphologically looking, differently looking cells, neurons, blood cell, muscle cells, okay, and so on. The heart cells. It means the differential gene expression which was kicked off and which made them different at the stage of cell fate determination, it is epigenetically inherited through successive mitotic divisions before cells became differentiated. When they start becoming visibly different after this early pattern formation, this process is called differentiation. They become totally different. And at the time of differentiation, they eventually leave the cell cycle. They no more divide. So this phenomenon, when a cell, when we are talking about a cell lineage, so this is the lineage of a cell. This is neuronal lineage, blood lineage, muscle lineage, heart lineage. The epigenetic inheritance means cells have memory, and we call this cellular memory. More precisely, transcriptional cell memory. At the level of transcription, genes are on or off. Each mitotic product of a mother cell remembers which genes are to be switched on and switched off, and how it does and achieves it is through DNA methylation, histone modifications. I'm not going into detail about non-coding RNAs, etc. But these are all these epigenetic modifications, which without causing any change in the DNA sequence, they enable a cell to differentially regulate genes. One of the key example we in our lecture slides we saw was X chromosome inactivation. What happens? Males are XY and females are XX. Now, it means the male cells, they contain only one X chromosome, which means genes which are present on X, as compared to female, there will be half the dose of the gene products. So females, they inactivate one of the two X chromosomes, and we call this X inactivation. At early embryonic stage, when embryo is, I think, fused cell stage, what happens? There's kind of counting mechanism, whether there's 1 or 2X at the molecular level. And one of the two X chromosomes, for example, we have X coming from father and X coming from mother. So it's a random process. It can be father's copy active or mother's copy active, father or mother. F means father copy is active and mother's copy is silent. Mother's X is inactivated in this baby. Here, mother's copy is active, father's is silent. So we call this phenomenon X inactivation, which involves, long non-coding RNA, as well as DNA methylation, as well as histone methylation. So H3 lysine 27 methylation, trimethylation is involved here together with DNA methylation. Polycomb group proteins are involved in silencing. The complete X chromosome, the whole X chromosome is switched off. And if we look at a cell which contains X chromosome inactive, it's like this is called bar body. The inactive X chromosome is clearly visible, whereas the active X is like a normal chromosome. Important thing is when this X inactivation takes place, now the progenitors of this mother cell, when this will undergo mitosis, both the daughter cells will keep father's copy active and mother's copy will be switched off. Here, the mother's copy will be on and father's copy will be switched off. Once gene expression state is established mitotically, then they are inherited through epigenetic inheritance. So this was about differential gene expression and its significance in mammals or in humans, and you can imagine what can happen. Imagine what can happen if a cell loses its transcription cell memory. Anyone would like to guess what can be consequences of losing transcriptional cell memory in the life of a cell? It can be cancer. Ji Amna.
I was gonna say that.
It can be disease. Because a cell, let's say a cell, my skin cell, this is differentiated. This is no more dividing. Imagine it loses its transcriptional cell memory that it committed with the skin lineage. It means different genes are being transcribed now, no more skin specific. It will enter into cell cycle and start uncontrolled proliferation, and we may end up having cancer. This is the significance of this subject. So let's go back to our favorite system, which taught us the most about how we develop. I intentionally first went through humans, but now I'm coming back to flies. Actually, this is where the cellular memory was discovered. Transcriptional cellular memory. This is where epigenetic factors, polycomb group, which are silencers, and trithorax group. So this is responsible for maintaining transcriptional cell memory of silent genes. This is activators responsible for maintaining transcriptional cell memory of active genes. Now, what they discovered that, yeah, Amna?
So this may be a naive question, but I wanted to ask, what's the difference between mutation and modification then? As in, isn't mutation
Excellent question.
naturally occurring modification?
Beautiful question. So when we say something epigenetics, it mimics a mutation. No. What mutation does, it normally switches off a gene. You lose function of a gene. And what epigenetic modification, for example, DNA methylation is doing, it's also causing loss of function. But we don't call this mutation because this is being done to temporarily lose its function, and it's having a beneficial effect if I lose function of a gene during development because it is not required in the development of that cell. Clear? So epigenetic modification is
Yes,
not
sir.
a mutation.
Thank you.
Good question. So let's see the fruit fly. Let's see what happens in our day one. Why we feel indebted to flies, why these little monsters, they come in our lives every day in 101 or even the whole biology curriculum. What they discovered that after fertilization, when female lays an egg, this egg already comes in a polarity, anterior and posterior, and dorsal and ventral polarity. What I'm going to teach you now in next 5 to 10 minutes is one of the biggest discovery of last century. The fruit fly, if you have seen its body, it's segmented. There are 8 segments in abdomen, there is head, there is thorax, and there's abdomen. They wanted to see how, this segmented body plan is established. They did a mutagenesis screen, random mutagenesis, and they found different mutants where segmentation was defective. And they started, in next 10 years, they learned that they have hit the jackpot, which was anticipated. What they discovered that there were certain genes, certain mutants, which showed a mutant phenotype only when mutation was in the mothers. We call them maternal effect genes. If mothers were mutant, father was normal. It means F1 heterozygous will be normal. No, because mother was mutant, they saw death. They saw strange phenotypes, completely abnormal flies. They named them maternal effect genes. They classified, categorized their mutants based on pure phenotypes, and they named them maternal effect genes, Gab genes. I'm writing them in an order now. Pair rule genes, segmentation genes, homeotic genes, and that's all. What they discovered that from this maternal genes, that mothers, when egg is still in the ovary, in the process of egg formation, mothers, they deposit certain messenger RNAs which are anchored at the anterior pole of egg or posterior pole of egg. And if one of these genes was mutant in mother, we saw this phenotype, death or severe abnormality, developmental abnormality. So while characterizing these genes, they discovered picoid, a maternal effect gene. What they discovered that picoid is present here, messenger RNA. And similarly, They discovered another one on the posterior side, and this is called nanos. They're anchored there. They're messenger RNAs. As soon as sperm hits, fertilization takes place. These messenger RNAs, they undergo translation. These messenger RNAs get translated and the protein diffuses. Remember, after fertilization, the fruit fly embryo grows in a very specialized way. The nucleus divides. So after fertilization, it becomes 2, 2 become 4, 4 to 8, and so on. Up to stage 13, I think this makes nearly 3 hours or so. They all these nuclei are sharing same cytoplasm. Cytoplasm up to stage 13. And all these nuclei, they are highly synchronous. They divide together. So same time. So you can literally see these mitotic divisions. This stage, when all the nuclei are at the same stage and share the same cytoplasm, is called syncytium. Syncytium. Or analogous is syncytial blastoderm. You remember blastula in there? Syncytial blastoderm. Now, what this syncytial blastoderm has to do with, we were talking about bicoid and nanos. We said we will have embryo, anterior, posterior. It got fertilized. Nuclei are there. This is a syncytial blastoderm. Cells keep dividing. This is bicoid. Why I'm showing you a gradient? Because the maximum messenger RNA of bicoid, it was anchored here, and nanos was there. So when they get translated, these proteins freely diffuse because it's the same cytoplasm. They diffuse, and as a result, we have a gradient. Cells here are exposed. If I draw X, if I try to draw a graph, let's say this is nanos, this is bicoid, anterior side, this is posterior. The cells which are here, they are exposed to only this much concentration of nanos. And let's say cells here, they are exposed to these cells are exposed to this much concentration of bicoid. As a result, what is happening? The result is we have cell population which is exposed to high bicoid, and cell population which is exposed to least bicoid. Similarly, we have cells which are exposed to high nanos and cells which are least nanos. Now, what is bicoid and nanos? Bicoid and nanos, these are basically transcription factors. They go within the nuclei, they bind onto, genes on these chromosomes, the DNA, they recognize their DNA binding sites in the promoter region and switch on. Here, a lot of bicoid responsive genes will be active. Same genes which are active here, they may be repressed here, but a different set of bicoid responsive genes will be on because they can respond to low concentration, and here a different set will be active. What has happened now? You have set up differential gene expression pattern there. In flies, there's no DNA methylation. DNA methylation is there only for first 30 minutes, and then it disappears. It's the histone methylation, histone acetylation, and different covalent modifications which play a role in epigenetic inheritance there. Yeah, there are two hands. Please ask question if there are questions.
अस्सलाम सर, मुझे बस पूछना था कि क्विज हो रहा है या नहीं, बिकॉज़ क्लास का टाइम खत्म होने वाला है and I have the next class.
या आज क्विज नहीं है.
Okay sir, thank you.
So the other question, there were two hands. दूसरे का भी
sir?
जवाब आ गया.
मेरा क्वेश्चन है कि you know how you said कि वो video में maternal genes, the mother basically provides the mRNA, hence there's a different gradient of proteins on the anterior and the posterior side, right?
Yeah.
Why is it like a gradient? Why doesn't it reach equilibrium? What's stopping it?
Very good question. It's a simple diffusion. At the source, you will have maximum concentration. And then, I mean, as the bicoid is moving from anterior to posterior, it comes across nuclei and it enters into nuclei. And within the nuclei, there is DNA. It binds to its recognition sites. So whatever does not go within the nuclei, which was here, is going to the next, to the next, to the next. And it's a simple diffusion principle. You take ink and you drop it in the bottle of water. It will take a long time before it can be uniformly distributed. But there is a reason nature hasn't done it uniform, because nature wants self-fate determination. Nature wants commitment of cell to different lineages. So purpose of having high bicoid here, a different set of genes will be active here as compared to different here, as compared to different there. And then mitotic products of these cells will commit to different cell lineages. Clear?
Sir, what about cells that can accept both the bicoid and the nano? Like, what happens to them? Is that possible?
Yeah, it is possible, but it depends on the concentration gradient. Concentration gradient, or we call them, so these nanos, caudal, etc. in developmental biology, we call them morphogen gradients, because they are involved in pattern formation. If we disturb the gradient, the whole body plan is disturbed. Clear?
Yes, sir. Thank you.
Now, important point is these maternal effect genes, which was bicoid, nanos, caudal, etc. Different names are coming to my mind. They are transcription factors, but they are transient. They remain there for 30 minutes or so, and then they disappear. So who is going to do their job then? What happens? Then comes the next class of mutants they discovered. Those mutants were named Gab genes based on the phenotype, and those genes are called Gab genes today even. They are also transcription factors. They are also transient. And what happens? They take over the job of maternal effect genes, and mitotically they maintain these gene expression pattern. The Gab genes, as compared to the maternal effect genes, they are not gradients. They are expressed in discrete regions now. For example, one of the Gab gene is Krupel. It's expressed like this. Then there's a Gab gene which is called giant. These are the mutant names. They were the names of the mutants. And the phenotype, based on the phenotype, they named them. And then later on, these genes were discovered. So Gab genes play their role as transcription factors. Then Gab genes also disappear as they are transient. Then comes the pair rule genes. Pair rule genes is the time where for the first time, and yeah, nobody of you asked me what happens after syncytial blastoderm. Do they ever become individual cells? Yes, after stage 13, which is nearly after 3 hours, these cells, all of them, they move to periphery. I forgot to tell you this earlier. And they become individualized. Now the cytoplasm divide, each individual nucleus, it has its individual cell membrane, and this is the stage called cellular blastoderm. Why it is important, why it came to my mind? Because at this stage, the first sign of segmentation, first ever sign of segmentation appears. An embryo is divided into 14 segments, clear segments. These are not brick walls, these are just cells, but they are grouped together. There's no physical barrier, cells are adjacent to each other, but we can see 14 grooves like this. And this is the earliest sign of segmentation in fruit flies. Light minor grooves. Okay. Now cells here, they were exposed to, now they are mitotically divided. You know, they were exposed to different morphogen gradients and Gab genes. At this time, when we have 14 stripes, there are 2 genes, even-skipped and Fushitarazu. These are the examples of pair rule genes. They are responsible for these 14 stripes. They are also transient. They disappear. They pass on the job to segmentation genes. Segmentation genes are the ones which eventually lay down the foundation for finally, you know, commitment to cell lineages. Different cell lineages are finally being finalized. And here, these are mostly cell signaling genes. Let's say wingless, notch, hedgehog. There's another transcription factor called engrailed, etc. They are involved here. And when the cell lineage commitment takes place, eventually the final class of mutants you remember we had here was the homeotic genes in their screen. What all of them are doing? What have they done? The maternal effect genes, they kicked off, they laid down the foundation for differential gene expression pattern. And this was carried forward by Gab genes, pair rule genes, and of course, organism was becoming more and more specialized. It was undergoing stages of gastrulation, then morphogenesis, eventually entering into organogenesis, etc. later on. But at this stage, when the embryo is still, I would say, 5 to 6 hours old, hardly, a special class of genes, we also call them selector genes. They are called homeotic genes because if a homeotic gene is mutated, you get one body segment transformed to another. For example, if the segment which is going to give rise to my legs, if there's a homeotic mutation in a homeotic gene, we may have legs on the head, or we may have instead of wings, we may have legs, or instead of legs, we may have eyes, etc. Okay, these are called master regulators of development. Homeotic genes, why I wanted you to learn about homeotic genes, they are conserved. Homeotic genes are conserved in flies, mammals, plants, and their function is to give symmetry to our body. What was shocking that homeotic genes, they normally exist on chromosome 3 in fruit flies in two clusters. One cluster is called bithorax complex, and the other one is called antennapedia complex. These are basically genes on chromosome like this, okay? And antennapedia, genes in the antennapedia complex, let's say this is antennapedia and this is bithorax complex genes. There are 3 genes in bithorax complex and there are 8 genes in an antennapedia. They go like this. Antennapedia, the first gene in an antennapedia, if you stain, it's expressed right on the most anterior part in the labia. And if you go from this side to there, the last gene on the homeotic cluster is called ABDB. It's expressed in the most posterior. This is not at all expressed beyond this region. And when we look at the expression pattern in mouse embryo, it's exactly same. It's not different. In flies, there's single copy gene, like there's a, let's say labial is a single copy gene, single gene is there. This is ABDB, which is in the bithorax complex. After ABDB comes ABDA, then there's UBX. All these 3, they make up bithorax complex. And then we have antennapedia, labial, deformed, proboscipedia, etc. These agents, they are expressed this way. If we look at the expression pattern, differential gene expression pattern, the way they are expressed in vertebrates, in mouse and in humans, it's exactly the same phenomenon. And these genes are conserved in evolution. And these homeotic genes, when they are active, since they are going to give identity to specialized cell types, they are involved in specialization of cells. Cell type specific gene expression pattern is due to these homeotic genes. These are transcription factors themselves. Now, it is very, very important that ABDB expression is maintained in all the cells here. All cells here and their future progenitors, they should keep ABDB on. And this is achieved through epigenetics by trithorax group proteins. They are H3, histone H3 lysine 4 trimethylating proteins. The ABDB will get its histones on the chromosome where ABDB will be. It will be H3 lysine 4 trimethylated by trithorax group proteins, and ABDB expression will be maintained. However, ABDB should be silent here. All here, ABDB should be off. And this is done by polycomb. UBX should be on only here. Trithorax group is going to keep UBX on here, but UBX will be off everywhere else by polycomb group. So this is all. This is about the epigenetic inheritance. Yeah, you may be surprised. These homeotic genes, they are conserved in plants as well. And the function of homeotic genes in plants is also symmetry, the pattern formation. Okay, so cell, specialization of cell types. And if you look at the example we had, they classified the mutants which were defective in flowering, the pattern of flowering. They classified them A class genes, B and C and B, and they discovered, eventually that these are homologs of homeotic genes, and their expression, if they get mutated, we get homeotic phenotypes. Any questions?
Excuse me, sir. I have a
Yeah.
question.
Yeah.
Can I ask? So like, I think since my FSC, I have the same question, and you like last in the last lecture, you mentioned that ke, after the formation of the embryo, the heart is the first organ which is formed.
Hmm.
Is it? So like, जो हार्ट को भी तो ऑक्सीजन चाहिए होता है, लाइक तब तक तो लंग्स नहीं बने होते, तो फिर हार्ट को ऑक्सीजन कौन देता है उस वक्त?
Heart को oxygen. So we get oxygen from blood supply, which we get from our mother. You know, if you look at the veins and the artery structure at the through placenta, you will be surprised at the level of engineering we have there. So we don't utilize oxygen or make our own respiratory apparatus. We are getting oxygen through blood supply, which we get from mother, and even the uh, hemoglobin, which we carry at that early stage, that the kind of stage you are talking about, is different than what we have today. Now we have uh, beta globin, but at that time we have different globin times. Did I answer your question?
Yes, sir. Thank you.
सोचने की बात यह है कि सबसे पहले हार्ट ही क्यों बनता है? Why not brain? All of you want to say, everyone says, मैं तो बहुत जो है ना सोच के फैसला करता हूँ, मैं तो मेरे से ज्यादा अच्छा फैसला कोई नहीं करता, because I sleep over my decisions and I बड़ा मैं दिमाग लड़ा. तो पहले ब्रेन क्यों नहीं बनता, हार्ट क्यों बनता है? Let me ask you today the final question. लाइफ तो ब्रेन से नहीं चलती या हार्ट से चलती है?
But sir, like brain needs the supply, like the heart supplies the brain with oxygenated blood and stuff. So isn't the heart
No.
more important than the
Look,
brain?
we don't have the heart there. If you have seen the ultrasounds, I was also shocked at that time when I first time saw first ultrasound of my baby. Uh, you know, it was few weeks after uh, the my wife conceived. I think hardly 4 weeks, and the gynecologist in Switzerland, she said, "Okay, come here," and it was all just beating. And when I saw the ultrasound picture, there was no heart. It was just a tiny, miny. The look at these Carnegie stages of development I shared with you in the lecture slides. It's hardly 3 weeks, and few cells which are drumming. They're just beating like hell. All you hear is tuck 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 tuck. There's this beating of heart very fast. So I wish we should have been seeing each other in the class, because I couldn't talk to you. So development brings you all very close to, मैं उसे कहता हूँ अल्लाह. आप लोगों में से कुछ जो अल्लाह को नहीं मानने वाले होंगे वो कहेंगे नेचर. What whoever we are, whichever religion we belong to, or we don't, even if we are non-believers, when you study development, it leads you to question many things that you ask bigger questions, much bigger questions, not differential gene expression, not how heart is formed, why heart is formed. You ask much bigger questions. And I often, when I teach development or differential gene expression, I always start with a line from Mia Muhammad Baksh Sahab. He was a Sufi शायर थे. He's buried in Kashmir कहीं, मीरपुर खास में शायद. तो उनका शेर मुझे बहुत वो स्ट्राइक करता है कि खसखस जितना कदर ना मेरा। खसखस मतलब खस कहते हैं स्ट्रॉ को, ये जो घास फूस ये जो गलियों में उड़ता फिर रहा है। तो खसखस जितना कदर ना मेरा ते मेरे साहिब नू वडियाइयां। मैं गलियां दा रूड़ा खूड़ा, मैं गलियां दा रूड़ा खूड़ा ते महल चढ़ाया साइयां। आई रिपीट, खस खस जितना कदर ना मेरा, कि मेरी जो वैल्यू है वो इट्स नॉट इवन लाइक स्ट्रॉ और फिल्थ। ओके, मैं गलियां दा रूड़ा खूड़ा, और वो गलियां कौन सी हैं? वो गलियां ये स्ट्रीट्स नहीं है, वो ये वाली गलियां हमारे बॉडी के अंदर, starting from our jugular vein all the way to our, where gametes are produced. If an oocyte fails to get fertilized, or a sperm fails to fertilize an oocyte, where it ends up? It ends up in a drain hole. So, मैं गलियां दा रूड़ा खूड़ा ते महल चढ़ाया साइयां। ये जो महल वो बना दिया है उसने, ये जो जिसे जो है ना सारे पे as soon as a baby is born, everybody is there to उसे चूमते हैं, उसे प्यार करते हैं. So when you study development, you ask much bigger questions. आप बायोलॉजी पढ़ते भी हो या ना भी आपके पास बायोलॉजी हो, आपको एक दफा हिलाती है. और ये तो the deeper you go in development, like genes, differential gene expression, ये तो फिर एक पूरी new universe है ना. It's a whole new thing and a fascinating area. अगर आप बायोलॉजी पढ़ने वाले हो या इंटरेस्टेड होते हो, अगर आपका पैशन होता है बायोलॉजी, then there's nothing better than going for dev bio specialization. आजकल तो जैसे stem cells, cancer, पता नहीं क्या कुछ है. So I wish you all good luck. If you have any questions, feel free to write me an email. Feel free to, ask Abdullah. If I'm not responding, ask Abdullah, that he has to bring answer from me, whatever your question is. Agar Abdullah, ताकि वो मुझे, he can, call me, he has my number. अगर मैं respond नहीं कर रहा, अगर if you are satisfied with Abdullah's answers, which I hope you are because he is an intelligent guy, तो या बाकी जो TAs हैं सारे, we have PhD level people teaching you in the as TAs. So I wish you good luck for your exam, and I hope to see you in Bio 100 lab maybe next week if you are in the Bio 100 lab, or in summers maybe. Allah Hafiz.
Good afternoon, sir. Thank you.
Allah Hafiz, sir. Thank you.